Hi, welcome. I'll be waiting for maybe 15 more minutes so that if there are others to join, they can join and then I'll start. Hi to the person who just joined. I'll be waiting till 6:15 so that if there are others to join, they can and then I'll start. मैंने ना स्टैंडर्ड PLC का है, तो उसका डैट फाइल आया। हाँ, तो उसमें क्या करना होता है, जो सीक्वेंस फाइल बना रहे होते हैं ना, तो जो फाइल नेम होता है ना एंड में, उसमें .dat लिखना है। हाँ, .dat लिखे। हाँ, हर एक फाइल नेम के एंड में .dat लिखेंगे तो डॉट दैट फाइल बनेगा। ठीक है, बस सबका आरटी अलग है। हाँ, ये मेरे को नहीं पता मैंने क्या किया है, बट ग्लूकोज का 14 मिनट के आसपास आ रहा है। ठीक है। हाँ, सब कुछ आगे बढ़ गया एक्सेप्ट एथेनॉल। एथेनॉल याद है पिछली बार 20 मिनट के आसपास आया था, इस बार 3 मिनट के अंदर आ रहा है। तो, तो क्या प्रॉब्लम है समझ में नहीं आ रहा है। स्टैंडर्ड बनाने में प्रॉब्लम है, क्या चीज़ है वो समझ नहीं आ रहा है। तो कल आ रही है क्या बाई चांस? हाँ, तो एक बार कल सिर्फ एक थोड़ा सा मैंने ग्लूकोज चढ़ा के देखते हैं, अगर मेरे स्टैंडर्ड बनाने में प्रॉब्लम है तो वो एक बार देख लेते हैं। हाँ, तो मैं एक बार देख भी लूँ। हाँ, और बनाया हुआ है, बट हम एक बार फिर से बना लेंगे, क्योंकि अगर मेरे उसी बनाए में प्रॉब्लम है देन वो देख लेंगे। और ऑलरेडी बफल है उसमें, तो आई थिंक कल सिर्फ 1 घंटा चलाना होगा। हाँ, आफ्टर रन भी कर दिया मैंने। हाँ, आफ्टर रन मैंने 4 आवर्स कर दिया। नहीं नहीं, मैंने डीसी मैम से पूछा था, अगर बफर चेंज हो रहा है देन 8 आवर्स इसकी वजह से नहीं कर पाएगा। तो हम बस एक बार वो करके देख लेंगे। अगर आज से तब भी अलग आ रहा है तो मैम ने बोला कि कल किसी और से बात करके नया कॉलम यूज़ कर रहे कि नहीं कर रहे, क्या चीज़ है वो एक बार पूछना पड़ेगा। हाँ, अच्छा, ये एक बार कल देख के डिसाइड कर लेंगे। चारू मैम का ये कि शायद कॉलम ही खराब हो गया इसलिए ऐसा आ रहा है, बट हमने 2 हफ्ते पहले किया तो तब तो अच्छा आ रहा था। तो हाँ, हाँ, हम्म, अच्छा, मैं सुबह आ जाऊँगी। All right, welcome everyone. Today is the week 3 of the problem solving session for the Plant Developmental Biology course. So we have 10 questions from the week 3 module, which will be similar to what you will have in your assignment, and then we'll discuss that. And if you have any doubts, please feel free to ask me. And whatever the question is, just write down the answer in the chat box. If you're not comfortable with unmuting yourself, you can unmute yourself and tell me the answer also. But you can, if you're not comfortable, you can put it in the chat box also. So, welcome. And I'm Anakha. I'm a PhD scholar in IIT Bombay, and I will be taking this problem solving session for you. So, let's start. All right. Uh, can someone just confirm if you can see my PPT? And it should be on the first question. Alright, so the question states that during root QC, that is quiescent center positioning, dash pathway is responsible for radial positioning of QC, whereas dash pathway is responsible for longitudinal position of the QC. So in this case, what they mean is that the differentiation pattern that you will observe radially and what you would observe longitudinally, right? What pathways are responsible for that? Okay, so can you write down in the chat box in case you know the answer, or even if you don't, you can just take a guess. Two, all right. Anyone thinks it's something else? All right. So yes, the answer is in fact two. It is the SHR, SCR pathway and the PLT pathway respectively. Okay. So let's try to understand how this works. So what you can see is a cross section of the root, right? And as you can see in this picture, okay, so radially, that is this way, okay, or this way, you are having some sort of differentiation of tissue. So what you can see is in the pericycle you have SHR mRNA being formed, which will produce the SHR protein, which is the red dot circle that you see here. That will go into the endodermis, okay, and in the endodermis you have SCR mRNA, which will produce SCR protein, and this SHR protein will bind to the SCR protein, okay, and it will stop its passage into the cortex, okay. So this will not, the protein will not move on to the cortex. So in this case, this is the wild type that you can see here, okay. This is scarecrow SCR mutant, and this is shortroot SHR mutant. Okay. So what you can see here is wild type. In the wild type, you have the cortex and the endodermis, right? Okay. But in mutants of the SCR and SHR variety, you don't have a differentiation like what you see in the wild type. Okay. So uh, in case you have SCR, that is scarecrow mutation, what you can see is that you have a area of, just 1 second. Sorry, I have a cold. Someone else has joined. We are on the first question itself. Okay. So as I was saying, yeah. So in the scarecrow mutant variety, you have a area of the green cells that you see are undifferentiated cells, okay, and they have characteristics of both the endodermis and the cortex. Whereas the short root mutant will have a area of cells that you can see in blue here, which will have the properties of the cortex itself. Okay. So, like you can see here also, in wild type, you will have endodermis and cortex. In short root, you will only have uh, cortex. In endodermis, sorry, in scarecrow, you will have a mix of both properties in the cells. Okay. So the question was during root QC path positioning, dash pathway is responsible for radial positioning, and that is HSR SER pathway. Now let's see how PLT pathway is responsible for longitudinal position of QC. So this is the longitudinal positioning of the QC. You will have the formation of root cap, apical meristem, basal meristem, elongation zone, etc. Right, and you have from the root cap, from wherever you have a primary meristem, you will have an excess of oxygen. So this area will have an excess of oxygen, and you will have a concentration gradient. So it decreases as you go up the root. Okay, and plethora is just one sec. Yeah. So it is a transcription factor. Plethora is a transcription factor. Okay, and it is dependent on oxygen. So if the amount of oxygen is high, the amount of plethora transcription factor will be high. As the oxygen is low, as it becomes low, the more you move up the root, the transcription factor plethora also becomes low. Okay, so in case you have a mutant of plethora, what happens is the stem cells lose their stem cell property. Okay, so to prevent that, you need to have plethora, and it is directly influenced by the amount of oxygen that is present in the cells uh, in the root. Okay, so the correct answer is obviously B in this case, SHR, SER pathway and PLD pathway. So I hope there is no doubt in this. In case you have a doubt, please write it in the chat. I'll go over that also. Uh, if there is no doubt, I'll move on to the next question. Okay. So please feel free to stop me whenever you have a doubt. Okay. So question number 2 is CLE 40 mutation results in, there is four options given to you. Option 1 is gain of stem cell activity in root apical meristem. 2 is loss of stem cell activity in root apical meristem. 3 is no change in stem cell activity, and 4 none of these. So what do you think the correct answer is? Anyone? Not sure about this? I don't see anything in the chat as such. No idea? Just take a guess, even if you don't know. Alright, 2. So the answer is in fact A. The first is the correct answer. So CLE40 mutation results in gain of stem cell activity in root apical meristem. So what you can see here is the QC, okay, and you have when you have CLE 40 mutation, what happens is instead of differentiating here, right? Instead of the QC differentiating into the cell, what happens is it turns more cells into stem cell like. So that is what you see here. So you will have more stem cells than in normal conditions. Okay. So when you have a CLE mutation, what happens is you gain stem cell activity in RAM. So you have the stem cells dividing and forming more stem cells and not forming differentiated cells. Okay. So as you can see here, CLE 40, okay, it will increase the amount of ACR4 CLV1, okay, which will inhibit WOX5. Okay, WOX5 is necessary for stem cell activity. Okay, so once it inhibits the WOX5 in cells, those cells can differentiate into the cells that you can see in this portion. Okay, the differentiated cells. But in a mutant where CLE40 does not work, what happens is this pathway will not work. So WOX5 is increased, as in the activity of WOX5 is increased because nothing is inhibiting it. Okay. So in that case, what happens is these cells, instead of differentiating, they will become more stem cells. Okay. So I hope this was clear to you. Does anyone loss of root meristem activity? Ah, okay. So that was a guess, right? So actually, it is gain of stem cell activity. It's a mutation. So what is happening is CL40 is not working in the mutation. Okay? So what CL40 mutation means is CL40 does not work in the cells. Okay? And in that case, since CL40 is responsible for this repression of WOX5, and WOX5 is responsible for the stem cell activity, what happens is if CL40 is not there, WOX5 works a lot better because it is not being inhibited and stem cells are formed. But if WOX5 was inhibited, those cells will differentiate. Okay, but what we need in this case is for cells to differentiate. So CL40 is important for that. I hope that was understandable. Uh, no doubts. Any doubts regarding any of this that I told you? Alright, so I'm assuming no doubt as such. Please feel free to stop me whenever you think you have a doubt, okay? And yeah, it's okay. That's perfectly fine, obviously. Anytime you have MCQ, it's best to guess if there's no negative marking, right? Um, Alright, so let's move on to the next question. During root development, the columella stem cells give rise to the columella as a result of asymmetric cell division. Okay? At later stages, the columella stem cells show the highest expression of dash, and the daughter cells, which is the differentiated columella cells, show higher expression of dash respectively. So you have option 1, clavata and mushel, option 2, scarecrow and shortroot, option 3, fez and sombrero, option 4, fabulosa and fabulota. So maybe you can write down in the chat if you know the answer, or if you have any guesses also you can write it down. Option 2, okay. Any other guesses? Option 3, anything else? Anyone think of something else? Alright, so the answer is in fact option 3. Okay, fez and sombrero is the correct answer. So let's see how that works. So what you're seeing is this slightly dark pinkish cells are the columella stem cells. Okay, and whatever is below them is differentiated columella cells. Okay, so what happens is in the columella stem cells, I don't know if you can see it in the image or not, but it will have fez in it. Okay, and till this is not divided columella stem cell. Okay, so what happens is just one second. Ah, okay. So this picture is not clear, right? So there is fez here also. Okay, so once the cells have divided, like you can see here, this is a columnar stem cell. Okay, and this is a columnar cell. So what happens is fez will increase the amount of SMB, okay, in the columnar cells, the differentiated cells. Fez will not be working at that point in the upper cell, which is the columnar stem cell. Okay, and once the differentiation is complete, Fez will not be there. Fez will not be present in the differentiated columnar cell, and Fez will be present in the columnar stem cells. Okay, so you initially, so just so that I have a little clarity. Okay, like in explaining, the first is you have columnar stem cells which will have Fez. When they will have an increase in cell size, but before division there will be Fez working in throughout the cell. Okay, but once the division has happened and you have one columnar stem cell and one columnar cell, the Fez is not working. Fez is there, but Fez is not working in the columnar stem cells, but the Fez in the columnar cells will uh, activate the SMB, okay, and the sombrero protein, that is SMB, okay, and it will eventually lead to your columnar cell having the activated SMB protein, okay, but not the Fez. The Fez is not working in it anymore, okay, but the Fez will be working in the columnar stem cells, okay, and again it can divide to form columnar cells and the same process continues. So in this case, this is the pathway that we're referring to. Okay, Fez will increase the amount of SMB, which is sombrero, but when you reach a certain concentration of SMB, it will inhibit the activity of Fez. Okay, so that is why in this case you had Fez initially. Okay, in the columnar cell, the Fez will be increasing the amount of SMB, but once you reach a certain amount of SMB, it will stop the activity of Fez in here, and that is what you see. In this case, since no SMB was formed, Fez will still be active here. Okay, so the correct answer in this case is Fez and sombrero. Okay. So at later stage, the columnar stem cells show the highest expression of Fez and the daughter cells show highest expression of sombrero. Okay. That is at this stage is what they're talking about. So Fez at later stages, the columnar stem cells show Fez and the columnar cells show sombrero. Okay. I hope that was clear to you. In case you have any doubt, uh, please feel free to ask me in the chat. If not, I'll move on to the next question. Okay. Alright. So the question is, which of the following transgenic will result in undifferentiated and multi-layered columnar cells in root? So option one is ARF10, ARF16 double mutants. That is plants which don't have ARF10 and ARF16. Fez mutant, miRNA 160 overexpression line. And option 4 is both ARF10, ARF16 double mutant and miRNA 160 overexpression line. So what do you think the answer is? Any guesses? Four. Yes. So the correct answer is in fact four. Both ARF10, ARF16 double mutants and miRNA 160 overexpression lines will have will result in a undifferentiated and multilayered columnar cell group. So that is what you see here. So the first plant that you see is the wild type. Okay. The second plant type is one which doesn't have the ARF10. The third is where it doesn't have ARF16. The fourth is where it doesn't have both ARF10 and ARF16. And the fifth is where uh, the microRNA 160 is overexpressed. Okay, so uh, those which don't have either ARF10 or ARF16 are still relatively normal, okay, like comparable to the wild type. But those which are either double mutant for ARF10 and ARF16, that means they don't have a working ARF10 and ARF16 gene, or overexpression of microRNA 160, show this sort of development where this sort of structure that you see, right, is because of undifferentiated and multi-layered columnar cells in the root. Okay, so uh, why does this miRNA in this case, that is overexpression of miRNA, why does it result in a similar phenotype? Is because overexpression of microRNA 160, what it does is it inhibits both these genes, ARF10 and ARF16. So in cells where you have overexpression of miRNA 160, it will act as a double mutant of ARF10 and ARF16 because this overexpressed miRNA is inhibiting the ARF10 and ARF16. Alright. So the correct answer was in fact ARF10 and ARF16 double mutants and miRNA 160 overexpression. So I hope there are no doubts in this question. Or if you have any doubts, please feel free to ask me. So ARF here stands for auxin response factors, right? And they are transcription factors. Okay, they will bind to any auxin response element. Okay, uh, in the promoter regions of these auxin response elements. Okay, so and these microRNAs are inhibitors of these auxin response factors. Okay, mRNA, this microRNA is small fragments of RNA, okay, 160. The 160 is telling you the type of miRNA in this case, okay. That is the name of this particular miRNA. Uh, and what happens when microRNA is formed is they will, there is gene silencing, right? Have you heard of gene silencing? So what these microRNAs will do, will they will silence the genes that are responsible for ARF10 and ARF16. Okay. So when you have lots of this miRNA 160 being formed, okay, which is microRNA, which is specific for this ARF10, ARF16 in this case. Okay. So they will inhibit the action of ARF10, ARF16 by stopping their translation, okay, of stopping the translation of these response factors. And since they are not being translated, they will not be present in the cell to, uh, help in the process, okay, that they normally do. Is it sort of clear, or a little bit more explanation? All right. So let's move on to the next question. So you have to select the correct order of following tissue with increasing levels of again microRNA 165A and 166B. Now here again microRNAs, the 165A and 166B are is giving you the different activities. Okay? Yeah. So you have to find out the correct order. Okay. uh, from the tissue which has the most level to the tissue which has the least levels of it. So from these four options, what do you think the correct answer is? This number that you see, the 165A and 160B, or the 160 in the case before, are just identifiers assigned to these microRNAs. Okay? Uh, During various experiments that you do and you figure out what these microRNAs do, you will assign them a number so that you can differentiate them from others. Okay? Just for clarification. All right. Two, you think the answer is anyone else? Yes. So the answer is in fact two. So endodermis has the most, then pericycle, then protoxylem, and then metaxylem. Okay. So these miRNA, 165A and 160D, they work against this HDZIP3 gene. Okay. So they are inhibitors, like I told you about the gene silencing, right? So they are gene silencers of this particular gene. So wherever you have high amounts of these microRNAs, 166 and 165, you will have less activity of this HD-ZIP3, and wherever you have less of this, you will have higher activity of this HD-ZIP3. Okay, so the endodermis will have the most activity of these miRNAs, okay, and the least activity of HD-ZIP3. And then there will be pericycle, which will have little less amount of miRNAs, then protoxylem, and the least will be in the metaxylem. Okay, and obviously, that means in the metaxylem, HD-ZIP3 gene will be active the most. Okay, so the correct answer is in fact endodermis, then pericycle, then protoxylem, and then metaxylem. Okay, does anyone have any doubts in this? No. Alright. So I'm assuming there are no doubts as such in this. Okay. So let's move on to the next question. The question is, which of the following protein is responsible for the nuclear degradation, specifically during sieve element development? So, like you must be knowing by now, sieve elements are part of the phloem, okay? And they are cells which don't have nucleus in them, right? So, somewhere along the development, they will initially be cells which have nucleus, normal cells, and then they will undergo this sieve cell element development, and then they will lose their nucleus, okay, because of nuclear degradation. Something similar happens to our red blood cells also, okay. So in the case of plants, which of the following protein is responsible for this? There are four options given to you: APL, CO, NEN, CUC2. So three, all right. Anyone else? So before I move on to the answer, just take into account the verdict. Okay, so the nuclear degradation specifically during seed cell element development. Okay, so this is the process that is ensured when you have enucleation of the seed cell element. Okay, so the answer is in fact NEN. That was option three, like you mentioned. Okay, so NEN 1 to 4 is responsible for the enucleation of the seed element. Okay, and this is a process that starts with APL, which will induce NAC45 and 86, which will induce NEN 1 to 4 for enucleation. Okay, and APL is altered phloem development gene. Okay, so in this case, specifically for since they are asking you specifically for sieve element development, that is why even though APL is in fact there somewhere in the process, since it is not directly used, since NEN 1 to 4 is directly responsible for initiation, therefore APL is not the correct answer in this case. Okay? CO is not the correct answer, sub 2 is also not the correct answer, and NEN is the correct answer in this case. It's a phloem cell. The C cells are in the phloem, not in the xylem. APL stands for uh, altered phloem development gene. Okay, I hope the APL was clear, like altered phloem development. Okay, all right, all right. So this is uh, a wild type plant that you can see, and this is NEN 422 mutant. Okay, so as you can see, in wild type, these cells are the seed cells. Okay, and they are supposed to have nucleuses here, but they don't. Okay, and the ones you see here also are in the stage of denucleation or enucleation, but where you don't have the N422 working properly, these seed cells will have the nucleus still within. So that is the proof for the fact of N124 working in enucleation for the cells. Any other doubts in this case? Uh, or Dinesh who asked, xylem cells are dead, right? This was in context with this question, or just in general? Alright, I'm not seeing anything in the chat as such. I'll answer in case I see it later. Okay. And then let's move on to the next question. So the question is, what would happen to the lateral roots of Arabidopsis thaliana if the IAA14 protein is stabilized? So uh, there are four options given to you. Option one is there would be no change to the lateral roots. Option two is lateral roots would be much longer. Option three is the plant would show no lateral root formation. And option four is lateral roots would be much thicker. So what do you think the correct answer is here? 3. Okay. Anyone else? Alright, I'm not seeing any other answer. So the correct answer is in fact 3. The plant would show no lateral root formation. So I IAA14 protein is a auxin inhibitor. Okay, so it is an inhibitor of this uh, response factor. Okay, so just one sec. Just have just one sec. I'm just okay. So related to the earlier question, that was cave elements are in the phloem, but protoxylem are living cells. Okay, and they are only present in growing areas of the plant. Okay, and they will differentiate into mature xylem cells, and that is the moment they will lose their cellular content. Okay, so protoxylem still have their cellular content, just for clarity. So what happened? So in this case, this is the question that we were looking for here, right? Option three. Okay. Anyone else? All right. So I don't see any other answer as such. So yes, the correct answer was in fact option number three, which was that the plant would not show any lateral root formation. Okay. So that is if IAA protein is stabilized. Okay. So IAA protein, IAA 14. Okay. So since it is IEA, indole acid, you might confuse it with an auxin, but it's not an auxin. Okay, it's a transcriptional regulator. Okay. So when auxin levels are low, this protein will interact with auxin response factor and it will repress the expression of the auxin responsive gene. Okay, this is only when the auxin levels are low. So that is what you can see here in this case, right? I hope the slide is visible, right? So that is what you see here. So in the first case, the OXIA gene will be responsible for the stopping of the transcription of this auxin-inducible gene. Okay, and in the second case, when there is enough amount of auxin, this OXIAA will not work, and the transcription of this auxin-inducible gene will be activated. Okay. So this is the wild type that you see here, which has lateral roots. These side may small roots that you see are lateral roots, right? But this is where you have an overexpression, or it can also be called a gain of function mutation of a IAA14. Okay, and in that case, what happens is that no lateral roots are present in this case. As you can see here, it's just one root. There are no lateral roots. That is because when the IAA14 gain of function happens, or like in the question said, when IAA14 protein is stabilized, okay, this oxo-inducible gene is not uh, transcribed. Right? In this case, it is not transcribed. Okay, and since auxin-inducible genes are responsible for the formation of lateral roots, no lateral roots are formed. Okay? So that is what you also see here. So auxin, high amount of auxin represses this IAA14. Okay, but when the auxin is low, this IAA 14 will repress the ARF7 and ARF19. ARF7 and ARF19 is usually responsible for the induction of these specific genes, the LBD16 and ASL18 genes, okay, which will in fact help you in the lateral root initiation. Okay, but when the auxin is high, okay, auxin will repress this IAA14 gene, so it is not able to repress the ARF7 and ARF19. So this will happen, this pathway will happen, and uh, the lateral root initiation will take place. Okay, but in case the auxin is low and the IAA14 is active, this entire pathway will not work and no lateral root initiation will take place. Okay? So again, you have a wild type here which has lateral root and a ARF7 ARF19 mutant, okay, which is a mutation in this part, not this, but it will show the same thing. No lateral root formation will take place. So, does anyone have any doubts in this question? I'm not seeing anything in the chat, so I'm assuming there are no doubts. So let's move on to the next question. So the question is, during lateral root primordia formation, following events occur. Uh, Nuclear migration, auxin response maxima, specification of founder cell, asymmetric cell division, and symmetry breaking. So you have to find out which is the correct order for these events, okay, in the lateral root primordia formation. So what do you think the correct answer is? Just take a guess. Two. Alright. Anyone else has any other answer? Alright. So yes, the answer is in fact two. So let's see what two states. Two states. The first thing is a specification of the founder cell. Okay. Then oxygen response maximum, symmetry breaking, nuclear migration, and asymmetric cell division. Okay. So that is the sequence. And let's see that. Okay. So in fact, the first step is the speciation of founder cell. So the cells which will give rise to your lateral root. So that is what when you specify those cells, they are called the founder cells. Okay. Then you have a auxin response maxima. So in the gradient of auxin that is present in the roots, you will have the maximum amount of auxin present in the cell at that particular time. Okay. And the high level of auxin will, like we saw in the previous question, will have this symmetry breaking effect. Okay. That means this cell, instead of being symmetrical in nature, will have, so that like you can see here, the nucleus will move to the lower part, okay, and the upper part, or in this case the nucleus will move to the upper part, and there will be a loss of symmetry within the cells itself. Okay. So symmetry breaking happens, nuclear migration happens. Okay. So the nucleus in both the cells move towards the center where they are joined together, and finally asymmetric cell division happens. Okay. So you will have in this case asymmetric cell division is because the cells are of two different sizes. So this will be a larger cell, this will be a smaller cell, and a larger cell. Okay. And these two small cells together, and they will still have a high amount of oxygen in them, will give rise to your lateral root primordia. Okay. Any doubts in this question, or the explanation, or anything like that? Alright, so I'm not seeing any doubts. So let's move on to the next question. After first asymmetric division of the zygote, the expression of dash gets restricted to the apical portion, and dash retains its expression in the basal portion. So you have one cell, right, and it divides into two, and the top one is called the apical cell, and the bottom one is called the basal cell, right? So after that division, what gene is restricted to the apical part, and what gene is restricted to the basal part? The activity, I mean. The gene will be present obviously in all the cells, but the activity is restricted to what part? So try guessing the answer. Any guesses? Alright, I'm not seeing anything in the chat. So I'm assuming that means you don't have a guess in this, but I'm audible, right? Alright, yeah. So the answer in this case is 3. So Vox 2 is restricted to the apical portion and Vox 9 is restricted to the basal portion. Okay. So let's see using a diagram. So this is the zygote, okay, and this is at a one cell stage, right? And you have Vox 2 and Vox 9, right? So after the first asymmetric division, that is what is here. So you will have a smaller apical portion and a larger basal portion, okay? And the apical portion will have Vox 2 and the basal portion will have Vox 9, okay? So this is a more straightforward question. Do you have any doubts in this question itself? As the division continues, the top two cells at uh, the top two cells will have the Vox2, then the cell connecting the apical portion and the basal portion will have Vox8 and Vox9, and the cells more south of that will have only Vox8. And in the 8 cell stage, again the top two cells will have Vox2, the next two cells will have Vox9, then the cell connecting the basal and the apical portion will have both Vox8 and Vox9, and the basal cells will only have Vox8. Okay. So since they're asking you about the first asymmetric division only, you have the correct answer as Vox2 and Vox9 respectively. Okay. So the top part has Vox2 and the bottom part has Vox9. Alright, so I'm not seeing any doubt as such in the chat. So let's move on to the next question. Question is, which among the following statements is false? DR5 is a synthetic auxin responsive promoter. Option 2 is auxin maxima is important for lateral root formation. Option 3 is clavata3 negatively regulates the expression domain of Wuschel, and option 4 is miRNAs do not play any role in shoot apical meristem maintenance. So, what do you think the answer is in this case? Any guesses? 4. Yes. So, the correct answer is in fact 4. So, uh, miRNAs do play a role in show typical meristem maintenance. Like we've seen a few examples before also, where they were playing a role in the root typical meristem maintenance, right? Similarly, they also play a role in the shoe typical meristem maintenance. So let's see what DR5 is in fact a artificially synthesized and highly active auxin response element. So that is what they were saying. DR5 is a synthetic auxin responsive promoter, which is correct. Okay. Auxin maxima is important for lateral root formation. We had seen that also. So you have auxin response maxima, where the cells, which are the founder cells, which will have a large amount of auxin present in them. Okay, and that will help those cells break the symmetry and have nuclear migration, and then finally asymmetric cell division before the lateral root formation takes place. So option 2 is also correct. Option 3 is clavata3 negatively regulates the expression domain of Wuschel. So let's see that. This is the Wuschel, okay, and this is clavata3. So Wuschel is responsible for induction of clavata3, okay. But once clavata3 reaches a certain amount or a certain concentration, it has a negative feedback loop where it will inhibit the action of Wuxia so that no more clevata 3 is formed. Okay? So option 3 is also correct. It negatively regulates the expression domain of Wuxia. Okay? That is what you see here. Negatively, uh, it is inducing. Sorry, it is repressing. Yeah. So it since clevata 3 is repressing the Wuxia domain, so option number 3 is also correct. So the correct answer in this case is uh, option number 4, which is false. mRNAs do play a role in the shoot effective mechanism maintenance. Okay, everything else is correct as in true. So that's it for today. That was the last question. Do you have any doubts regarding any of the questions taken today? Okay, so enucleation means you have a cell with a nucleus, right? So what happens during enucleation is the cell, the nucleus in it is destroyed. So this diagram might not be good for that. So you have a cell with a nucleus. After enucleation, the cell will not in fact have a nucleus. Okay? So the same thing happens to our red blood cells. So you know red blood cells have this structure, right? If you see it from the side. Usually cells have this structure in our body because in the center the nucleus will be large and you will have this sort of curve that you see because the nucleus is here. But in RBCs, because they have undergone enucleation, whereas the destruction of the nucleus is death, we have this structure. Did you understand what enucleation means? Yeah, alright. Any other doubts? Usually anything you add E in front of usually means a removal of. Okay. So enucleation, nucleation के आगे if you put E, that means removal of nucleus or removal of nucleus. Okay. Alright, I'm not seeing as such any doubt. If you don't have any doubt, you can leave the meeting. That was the last question anyway. So if you don't have any doubts, you can leave the meeting. If you have any doubts, you can stay back and I will clear them for you if I can. Divya, do you have any doubt? Alright, I'm going to stop this meeting. Uh, do you have any doubt, Divya? Or if not, I'm just going to stop this meeting. Alright.